Not many people here this morning. Not sure. I'm sure next Tuesday morning at 8 there will be a lot more. So yes, 8 a.m. Tuesday morning. 8 a.m. Tuesday morning, 8 a.m. Tuesday morning. How many times do I have to say it? Yeah, so that's when our final will be. We'll have a review session on Friday. No clicker questions on Friday. I'll try to have some today. No, I'm not George Kaysen, my graduate student, because he's not giving the guest lecture today, but I'm actually kind of psyched because I haven't talked about CRISPR in a long time, and so this will be really kind of fun. So first ever wanted to finish up talking about post-transcriptional regulation, and basically this set of bars here is what we haven't talked about yet. We haven't talked about nuclear export, localization, and basically translational regulation. So last time what we didn't get to was nuclear export and the regulation of nuclear export. So again, basically anything that happens, you have transcriptional initiation, but everything after transcriptional initiation can also be regulated. Capping, splicing, tailing is what we talked about last time, and editing. We can also regulate export. And the best understood regulation of export has to do with HIV-1 and a number of other retroviruses, but best understood from HIV-1. So HIV-1 is a fascinating virus. We'll spend a whole lecture talking about it next term in virology. But two sort of things happen with HIV. You've got spliced RNAs, which are being made into HIV proteins, and then you have the HIV genome, which is a whole unspliced piece. And the HIV genome needs to be exported from the nucleus. So normally, and everything we've talked about so far has basically said unspliced RNAs don't get exported because they don't have all of the appropriate machinery that's associated with them. But in the case of HIV, that genome does need to be exported. And the way that that works should sound really familiar now, is we have a particular element. So element, again, this is a nucleotide sequence present in the RNA. These elements interact with proteins. That particular protein is called Rev. The name is not that important. Binds to this Rev response element and transports the unspliced genomic RNA out into the cytoplasm, where then it gets packaged and made into virions that will go off and infect other cells. There's other kinds of regulation of export as well, most of them, again, having to do with a specific sequence in the RNA and a protein that's going to interact with that sequence and pull it out of the nucleus. Once it gets pulled out of the nucleus in the case of HIV, this now is going to serve as the virus genome and get packaged. But there are other things that happen to messenger RNAs after they get out in the cytoplasm as well. And we already talked about this when we talked about the case for regulation of genes, particularly those early development genes in Drosophila. So you have gradients of these regulatory proteins. Bicoid and hunchback, the positive regulator is important for the even skip gene in Drosophila. How do you get these gradients in the embryo? The way you get the gradients in the embryo is that the mRNA that encodes those proteins is stuck at one end of the embryo. So actually is literally transported to that end of the embryo. And that's basically shown here. You have a cell. In this case, we can say it's a Drosophila embryo. And then a number of proteins up here at the end. Again, in the case of bicoid and hunchback, this would be the anterior end. Messenger RNAs are exported from the nucleus. Then they somehow need to get to this end. And there's lots of different ways that that can happen. It's all dependent on the 3' end of the messenger RNA. And we haven't talked too much about this yet, but if you think about messenger RNAs, you've got a 5' end, you've got a 3' end, you've got the coding sequence in the middle, but you also have bits of the RNA which don't code for protein. And those are, again, incredibly creatively named untranslated regions, or UTRs. Yeah?
Were we supposed to splice those out?
So the 5' and 3' ends you still have. The exons you have, the introns are gone. But the very ends, between the cap and the start, and between the stop codon and the poly A tail, those you still have. And it turns out those are really important for regulatory purposes. So those you still have, and you kind of have to have them. There's the cap, and when we have you have to think about translational initiation. Cap binding by small subunit of the ribosome scoots along until it hits the start codon. That region that it's scooting along, that's the 5' untranslated region. Then you have your stop codon, and then after that you'll have where CTSF stimulates cleavage and poly A tail formation. That's where you have your 3' UTR, untranslated region. And this is, I'm spending some more time talking about untranslated regions because it's going to be really critical for half of today's lecture. So these 3' untranslated regions, just like we had just talked about for getting export, also have specific sequences in them. Those sequences are going to interact with various different proteins. In some cases, it's going to be a protein which is going to allow these to be transported along actin filaments to one end of the cell. In some cases, it's just a binding site for whatever proteins there are at one end of the cell. Or even in some cases, you have degradation, and we'll talk more about mRNA degradation later on, that happens to all of these RNAs unless they happen to be protected by being bound at one end of the cell. And so people also talk about these 3' UTRs as zip codes. So the cell knows where to deliver this particular messenger RNA. And again, it's all having to do with proteins that are binding to specific sequences in those untranslated regions. So we have mRNA transport, mRNA localization. Now what comes next? After you've got all of your messenger RNAs, what happens next? Translation. So hopefully we'll have translation. So translational control, again, just like all of these other things, is also highly regulated. It's going to be a little bit different in terms of excuse me, bacteria versus eukaryotes because translational initiation is different in these. So translational initiation, great review for the final. In bacteria, you have the small subunit of the ribosome through base pairing interactions finds the start codon to that Shine-Dalgarno sequence. And in eukaryotes, as we just mentioned, binds the cap and scoots its way along the RNA. So if you want to regulate translation in bacteria, you want to regulate the small subunit binding to the Shine-Dalgarno sequence, which is always right next to your start codon. There are a number of proteins that will bind to sequences and very often structures in your RNA that will block the binding of the polymerase. So this is a regulatory mechanism where in the presence of this repressor protein, you're not getting any kind of translation. We talked a little bit about riboswitches before in terms of translational termination, transcriptional termination. This has to do with translation. And again, it's an aptamer, a small RNA sequence that can bind to something. And here, it's binding to a small molecule that causes a change in the secondary structure where you can't bind to the AUG. When I say you, I'm meaning the small subunit of the ribosome. So these are two possibilities. My favorite is actually this one here. It's also called a thermometer. And this is literally a way that you can regulate translation just based on temperature. So way back when we talked about hybridization and heating up DNA or RNA, the two strands come apart, you cool it down, they come back together. So this particular case, there's a secondary structure that forms in the RNA at a lower temperature. You heat it up, this secondary structure melts, and you have access to your AUG. You cool this down, those form back up, and you have a secondary structure so you can't get binding by the ribosome anymore. So incredibly simple and elegant system. because all it is dependent on temperature, and not surprisingly, these genes that are being expressed in this case are heat shock genes, like chaperones that need to be turned on at higher temperatures because proteins aren't folding properly. They're falling apart a little bit. So this is a really nice way of regulating, also a very fast way of regulating. The messenger RNA is here and then can be unfolded. And then the last case, and we're going to talk more about this as we go on, is just making a separate RNA or an antisense RNA. And so antisense RNAs are going to just bind base pair to the ribosome binding site. And instead of binding the ribosome binding sites, the ribosomal RNA can't bind there anymore. And so the expression of this short antisense RNA is also going to be shutting down translation. And we'll see one of the really nice things about these short RNAs, which again will become important when we talk about CRISPRs a little bit later on, is that base pairing interaction is incredibly specific. Again, we talked about this when we talked about hybridization for DNA, but the same thing is true for RNA. You'll find all of these base pairing interactions that can then be a very, very specific one. So this is in bacteria. The translational initiation in eukaryotes is, again, it's binding to the cap and scooting along your messenger RNA. So none of these things would actually work in a eukaryotic system. But there also are things that you want to regulate in eukaryotic translation. So the first one of those that I wanted to talk about is regulation now not of the RNA, or for that matter, the ribosome binding, but regulation of getting the initiator tRNA associated with the ribosome and releasing it when you have association with the start codon and the large subunit comes in. So normal translation, association with start codon, hydrolysis of GTP by eukaryotic initiation factor 2. That allows the initiator tRNA to be incorporated into the ribosome, and you can start translation. Well, one of the things that we didn't talk about before is how you get GTP-bound eukaryotic initiation factor 2. Just like, again, we talked about way, way, way back, we have nucleotide exchange factors. This is a classic G protein. When it's bound to GDP, it's not active. GDP has to come off. GTP has to be put on. How that happens is AF2B. I don't expect you to remember the name, but it's a nucleotide exchange factor. And all of these GTP binding proteins have to have these nucleotide exchange factors. So putting on GTP. So this is what happens after you have translational initiation. You have new EIF2 bound to GTP. This goes off, finds a new initiator tRNA, brings it back into the ribosome. However, if you have phosphorylated EIF2, this guanosine nucleotide exchange factor gets stuck. It can't exchange the GDP for GTP. And if you can't exchange GDP for GTP, your initiation factor is never going to be able to bind an initiator tRNA. You're never going to be able to start translation again. And we've talked about translation being energetically very, very expensive for the cell. So these are the kinds of things that happen under stress conditions. And the best understood stress condition is, oh, I just got infected by a virus. So that is one of the times that you have phosphorylation of EIF2. Another reason you would want to stop translational initiation if you've just been infected by a virus is that you don't want to be translating all the virus proteins, do you? So let's shut down translational initiation until we can get rid of that virus. So this also seems to be what happens when you have a quiescent cell in the G0 phase, and it's all dependent on these phosphorylation, protein kinase, which is phosphorylating this EIF2. So shuts down translational initiation. So this is clearly a problem for viruses because they would like to have their genes being translated, or our messenger RNAs being translated, again, to totally over-anthropomorphize here. So some of these viruses have come up with this really fascinating solution to this problem. And it's called an iris. What the heck is an iris? An iris is an internal ribosome entry site. So instead of the ribosome associating with the 5' end of your messenger RNA, this can associate somewhere in the middle. And so that's what these irises are. Again, internal ribosome entry site. Internal ribosome entry site, basically this provides a platform to get all of your other translation factors to come and bind to this messenger RNA in the absence of cap structures and the proteins that are associated with caps. There are lots of viruses that use these internal ribosome entry sites. Zika is a really nice example of these, but other viruses use them as well. Just like we saw with splicing, splicing was thought to just happen in viruses. Turns out this happens in cellular proteins as well. So it's not just an virus-specific thing. It's also present for cellular messenger RNAs as well. So we've got translational regulation in terms of blocking initiation, which is phosphorylating of EIF2. We can get initiation in different places, a cap-independent translation with iris. But then a third thing in terms of translational regulation has to do with messenger RNA stability. So we've talked about the polysomes, where you've got this nice circular messenger RNA. The 5' end and the 3' end are associated with each other because you have the EIF4G forming the bridge between the poly A tail and the cap binding complex. So one of the reasons you have that is that the ribosomes can recycle very easily. Another reason that you have that is that it stabilizes your messenger RNA. And stabilization is really all about protecting against the hungry exonucleases that we just love to chew in at the ends of your RNA. So the cell has taken advantage of this in terms of if you have a messenger RNA which is stable, that can continue to be translated for a long time, you make a lot of protein. However, if you want to have a messenger RNA that doesn't make too much protein, then one way to control this is have a messenger RNA that gets broken down and broken down relatively rapidly. So at the top here, we have examples of, and this is not going to work anymore, a normal messenger RNA, which relatively slowly, these poly A tails get chewed off. Once the poly A tail gets to a relatively short amount, this is probably where you don't have binding by the poly A binding protein anymore, then there's endonuclease, which chops in the middle of your messenger RNA, at which point you have really rapid degradation of your messenger RNA. It gets chewed up by all of these exonucleases. So again, this is what happens. Normally you've got a relatively stable messenger RNA, and these messenger RNAs in eukaryotes are way more stable than the bacterial messenger RNAs. And if you think about bacterial messenger RNAs, again, nice review thinking about translation. As messenger RNAs are being made by bacterial polymerases, RNA polymerases, they're being translated immediately. So messenger RNA comes out of the RNA polymerase, ribosome binds onto it, and starts translating it. What that means is you also don't have any protection at the 5' end of your messenger RNA in bacteria. And often that starts to get chewed off because there's no caps in bacterial messenger RNAs. And so bacterial messenger RNAs are actually really pretty unstable. On the other hand, eukaryotic ones are very stable indeed. So if you want to have something that breaks down faster as a eukaryotic messenger RNA, often you have some kind of binding site, and in this case, literally a endonuclease site. In this, comes back again, 3' UTR, the light blue stuff. So again, 5' UTR over here, 3' UTR over here. As soon as you get rid of that poly A tail, which is what happens when you have endonuclease, then this gets broken down incredibly rapidly. And it's not just from the 3' end it gets chewed in, it also gets chewed in from the 5' end as soon as you get rid of the cap structure. And there are specific decapping enzymes that are going to be stimulated as soon as this messenger RNA starts to get degraded. So let's look at a couple of examples of these. Try and get this to work again. First of these is a regulation of two actually different RNAs, and they're regulated by a binding protein. And actually, these are the same binding proteins in the 5' UTR and in the 3' UTR. Again, most of these RNA binding proteins, they don't bind a single-stranded RNA, they bind a double-stranded RNA, and that double-stranded RNA is formed as some kind of secondary structure in order to have these binding taking place. The protein here is called aconitase, and yes, it is an enzyme, but we only care about it now because of its RNA regulatory mechanism. Aconitase regulates two specific genes, a gene called ferritin and transferrin, so ferritin and transferrin. Ferritin is a protein that will bind to iron and actually sequesters iron, because if you have too much iron inside the cell, iron is very reactive, and particularly with something called the Fenton reaction, which can generate active oxygen species, big problem. So if you got too much iron, you want to make ferritin to block all of this iron from doing things that it's not supposed to be doing. On the flip side, we do need iron for actually quite a lot of our enzymes. So if you have no iron, you need to bring iron inside the cell. And that's what the transferrin receptor is all about. So it turns out it's just one protein, this aconitase protein, that binds to basically an almost identical sequence in both the 3' UTR of the transferrin receptor and the 5' UTR of the ferritin gene. So binds in both these places. If you have extra iron around means that the aconitase is no longer binding to either the 5' UTR in ferritin or the 3' UTR in the transferrin receptor. What happens is if you have binding of this protein, this secondary structure allows you to get translation here, but it blocks translation over here. If you have no aconitase binding, you make the ferritin protein because it's being translated. You don't make the transferrin receptor. And so this is also, in some ways, you can think of this as being kind of analogous to what happens in some of those bacterial regulators, transcriptional regulators. Presence of a ligand is not binding, and in the absence of a ligand, it binds. So just like you see with a lac repressor. So a lac repressor binds in the absence, comes off in the presence of. So that was just one example of these regulations. But again, it's mostly about proteins interacting with a RNA sequence, and that RNA sequence is almost always in some kind of secondary structure. So just to review what we've talked about in terms of post-transcriptional regulation, we had our riboswitches, which were the aptamers that bind to a specific ligand. And in the case of the first riboswitches, we talked about them in terms of getting transcriptional attenuation, so premature termination. Talk a little bit about alternative splicing and how that's regulated. The example we used there was the sex-lethal protein. And quick aside, someone was asking me some questions about the sex-lethal protein on my email, and I've posted a couple of discussions about that on D2L under the discussions, questions, and answers. Alternative tailing, this is our antibodies. If you have high amounts of your cleavage and stimulation factor, you get cleavage of your messenger RNA and tailing that takes place at a different place than if you have a lower amount of CSTF, and this turns out to be exactly what happens in B cells. We have antibodies that are either bound to the membrane because they have a hydrophobic tail to them or are being secreted. We talked a little bit about RNA editing. This adenosine deaminase that's dependent on double-stranded RNA, which will modify A's to I's and give you recoding of your sequences. There's also C to U's, which are being functional. And today we talked more about where you get your messenger RNA, i.e. getting it transported out of the nucleus, getting it transported to a particular part of the cell, and then translational control from the point of view of having binding to different parts of the RNA to block either the stranded RNA sequence, so getting the small segment of the ribosome, or blocking it in some way of some kind of binding protein that's associated with it. And I thought that I had a clicker question here, but it looks like I don't. What did I do with it? Drat. Okay. So maybe there's some more later. Maybe I just put it in the wrong place. We shall see. The problem is when I save these things and post them on D2L, I have to change the presentation, and then maybe I didn't save that previous presentation, the one that still had the questions in it. I'm giving away all my secrets. So just as an overview of the last part of the last new lecture of the term, and I'll put those questions up and just give you the answers. So maybe I should just give people 2 points for everybody for just being here. Actually, the last question was one I was going to give everybody a point for anyway. So I'll just give everybody 2. And you don't even have to be here to get them, because I don't know everybody who's here. Actually, I don't know all your names quite yet. So what's going on with RNA? And again, we could have a complete course on RNA and all the cool things that RNA does that actually has absolutely nothing to do with messenger RNA being just this mechanism to go between DNA to protein. So we've talked a little bit about some of the ribozymes already. Self-splicing introns were the first ones to be found, but the ribosome is also a ribozyme. There are a number of other ribozymes that have been well studied. RNase P, so this is a RNase which is really important for processing tRNAs, also has an RNA component, and that RNA component is active. But I wanted to talk today about regulation of gene expression, because it turns out that there are lots of RNAs which are involved in the regulation of gene expression. So first of those are the long ones. We've got long ones and short ones. Again, these molecular biologists, unlike the Drosophila geneticists that have a huge amount of creativity in terms of naming, the molecular biologists have much less so. So long non-coding RNAs, that basically tells you exactly what it does. And what do we mean by long? Long is more than 200 nucleotides. And in this case, these are non-coding RNAs that just the RNA has an activity. When I say just the RNA, no proteins, well, actually these long non-coding RNAs almost always associate with proteins in terms of getting their different activities. So there's a couple of different ways that these things work. And in fact, at the top here, I should mention, at first people thought there were thousands of long non-coding RNAs. There may actually be tens of thousands of long non-coding RNAs. It's actually really hard to tell whether a particular RNA is actually serving as a regulatory RNA or not. And the more that we look, the more that we find in terms of these things. So these long non-coding RNAs can either base pair with RNA, and we kind of talked about this in terms of antisense RNA. So if you got antisense RNA, the long non-coding RNA can bind to it and then block translation. But it turns out that this kind of long non-coding RNAs binding to another RNA can actually take that RNA and put it somewhere else inside the cell. So not unlike the case we talked about for the Drosophila location of messenger RNA to one end of the embryo, can actually sequester these messenger RNAs in subcellular, or even in some cases subnuclear compartments. They can also interact with DNA, so these long non-coding RNAs, and those are the examples that we have over here. So 2 of these, one of them is you have a long non-coding RNA being transcribed by cellular RNA polymerase that actually binds to proteins that will then block expression of genes that are near to it. Now, so how does that work? It turns out that a lot of these are binding to chromatin remodeling enzymes that could either be histone acetyltransferases, methyltransferases, etc. So just the presence of this long non-coding RNA together with these proteins can block activity, and even some of these long non-coding RNAs will be transcribed in one place, and then, due to the RNA binding sequences it has here, go and act in trans on a completely different chromosome. So it's not just a long non-coding RNA that's being made in one place that's going to be acting on genes next to it. It could also be released and then act in a trans form. Most of these long non-coding RNAs here, they're shown as having lots of different proteins that are bound to them. It does seem to be that they are binding to these proteins, and it's moving these proteins around, which is giving you a particular activity. Now, so where these proteins are is going to be dependent on where you have the base pairing interactions between part of that RNA and some DNA or some RNA in another place. So it's the RNA itself is providing your specificity. The activity is coming from the proteins. And we talked about this when we talked about the snow RNAs that I think somebody else asked me about as well online. Small nucleolar RNAs, they cause modification of ribosomal RNAs. The modification takes place through proteins that then are brought to a specific place due to the snow RNA. So specificity from the RNA, activity coming from the protein, and again, that seems to be happening here as well. So long non-coding RNAs, so those are the big ones, above 200 nucleotides. What about the ones that are smaller than 200 nucleotides, or the small RNAs? This is also very often put into the overarching group of RNAi or RNA interference. So what happens with RNA interference and many of these small RNAs, you start out with a double-stranded RNA. Now, usually you don't have much double-stranded RNA inside a cell, and this was thought actually to have originated as a defense mechanism against viral infection. So all RNA viruses end up with double-stranded RNA as part of their replication cycle. And this is now actually two molecules of RNA. So it's not some kind of secondary structure that's formed in your RNA. It's literally two strands that are complementary to each other. If there are two strands of RNA that are complementary to each other, you have specific proteins that will recognize these double-stranded RNAs and chop them into short pieces and then get rid of one of the strands. And they're bound to these proteins which are called either Argonaute proteins or PB proteins. These proteins now can go and find a different RNA, but it's the one that matches the same RNA that you have over here, and can do a number of different things with it. The classic example is chopping up that RNA, because again, this is likely to be a viral RNA, likely to be an RNA that the cell doesn't want to have around, so it gets chopped up. You can also have these small interfering RNAs, that's the RNAi, RNA interference, which lead to just translational repression. And translational repression, just like we talked about those antisense RNAs in terms of translational regulation that you have in bacteria. They just bind double-stranded to your messenger RNA, and that double-stranded messenger RNA can't get translated anymore. And if it doesn't get translated for a long period of time, eventually it gets broken down. And there are a few cases where this double-stranded RNA form, again, together with these proteins, leads to heterochromatin formation. We'll look at some examples of that a little bit later on. RNA interference was originally found in plants. Plants are infected by a large number of RNA viruses, so it's not terribly surprising that there's a nice, strong RNA interference mechanism here. But it was found in a really interesting way. There were people who were trying to make a really purple flower. And so what they did is they took some extra RNA for the purple gene, and they injected it into the flower, and instead of it becoming more purple, it went white. It's like, wait a minute, you put in, RNA, it's supposed to, this RNA is supposed to be giving you, more of this purple gene color. No, completely shuts it down. And then a similar thing was found when people were looking at RNA genes that were regulatory in C. elegans. Same thing. You put in extra RNA, and those extra copies of RNA, instead of stimulating expression, because they thought that was going to happen, actually ended up suppressing expression. And so, long story short, this led to the Nobel Prize in 2006, just by, if you just take double-stranded RNA and inject it into a cell, you can then shut down expression of genes that match that double-stranded RNA. Turns out to be incredibly useful, and we'll take a look at an example of that a little later on here. So that's RNA interference. And this is great, works well against virus infection. But it turns out that this is all external RNA which you're putting in. It turns out that there are also internal RNAs that are encoded in our genome that also function as short RNAs. So these are what are called the microRNAs. MicroRNA is, again, short RNA. These are encoded in genomes. These are made as a single RNA that has a lot of secondary structure that's associated with it. This secondary structure is then chopped. It's also called a cropping mechanism. And then it's diced, where it's chopped into an even shorter length. And then this shorter piece gets associated with this Argonaute protein, which is exactly the same protein we talked about in terms of RNAi before. Now we have Argonaute plus one of these short RNAs, known as a RNA-induced silencing complex. If this has an exact match to a particular messenger RNA, it gets chopped up. If, however, there's a less extensive match, and this is what seems to happen in most of these microRNAs, part of your microRNA will bind to the messenger RNA, but part of it won't. And this leads to translational suppression. And very often, this is going to be binding again in our friend, the 3' UTR. So 3' UTR has the binding site for a microRNA. And again, critical here is that these microRNAs are made by normal genes inside the regular genome. So these are transcribed, made by the normal translation, it's like transcription machinery, they don't get translated. The transcription machinery then processed and used for gene regulation. There are literally hundreds of these genes, and actually probably thousands of these genes as well, that are present in our genome. And again, they're interacting mostly through the 3' UTR. Just as kind of a reminder here for these small interfering RNAs, these are exogenous. So they're double-stranded RNA that's coming from somewhere else. They're not a specific gene that has this structure that gets then chopped up. Interestingly enough, these double-stranded RNAs get chopped up by the same protein, dicer, that also chops up your microRNAs. But now these are double-stranded RNAs which are going to match this double-stranded RNA that you had up here in the first place. Here interacts with these Argonaute proteins, RNA-induced silencing complex, chops up your RNA if it matches completely. But these small interfering RNAs can also be involved in translational silencing through a different set of proteins. So where would you have these double-stranded RNAs? Again, I mentioned before, this can be double-stranded RNAs that come in from RNA viruses, why they were found in plants in the first place. But also, these double-stranded RNAs seem to also work to get rid of the activity of transposons. So we all remember, maybe or maybe not, about 40% of our genome are retrotransposons. If 40% of our genome's retrotransposons were being transcribed and jumping all over the place all the time, this would be a big problem. How do those move? They move through reverse transcriptase and endonuclease. Those have to be transcribed and translated. If you've got lots of copies of these genes all throughout the genome, and they're more or less identical to each other, if you have transcription over here of one of those transposons, transcription over here of one of those other transposons, you're going to end up with double-stranded RNA which matches the transposon sequence. Double-stranded RNA that matches the transposon sequence, that turns on some of these mechanisms which can then shut down the transcription and translation of these transposons. So it'll chop up the messenger RNA or make sure that messenger RNA doesn't end up getting translated. You also end up with double-stranded RNA in centromeres because these are highly repeated sequences. Again, they're being transcribed. So you end up with a region of the genome where you also suppress any kind of transcription, and part of that is through these small interfering RNA mechanisms. So why do we care about this? A, it's protecting against virus and transposons, but it turns out these are also really, really useful for various applications in molecular biology. So if you want to turn down a gene at a particular time in your experiment, very often you use a small interfering RNA. And this is literally from an email that I got yesterday in my inbox, selecting the right siRNA for your experiment, tools for in vivo siRNA, which controls should I use for my siRNA, and which gene editing tools are right for me. So this is a very, very active area of research, and then using all of these small interfering RNAs. So for gene function, it's great. Medical applications, not so good. So there are a number of companies actually started by, among other people, people who got the Nobel Prize because they discovered RNA interference. So those turn out to be, bringing these small RNAs into cells and using them for suppression of transcription and translation just really hasn't worked very well. And it's not entirely clear why that's the case. RNA is not terribly stable, but for some reason it's not, hasn't worked terribly well. And really it's a lack of efficiency. And then there's this other aspect right here, which we'll talk more about when we talk about CRISPR in just a second, is the idea that you can have sometimes some interaction which is actually not the interaction that you want to have. So as I mentioned, for slicing and dicing up of your RNAs, that's if it's a perfect match. But if you have an imperfect match, that can shut down translation. But if you have an imperfect match, so you've maybe got a perfect match somewhere in the gene that you want to get rid of, but now you have an imperfect match somewhere else in the genome, that may be shutting down translation of a gene that you really don't want to be shutting down translation of at all. So becomes this really kind of tough balance, and why this really hasn't gone anywhere with medical applications. So now I'm going to switch gears. We'll spend the last 20 minutes or so talking about CRISPR. So everybody know what CRISPR stands for? Down here on the bottom of the slide. Clustered, regularly interspersed palindromic repeats. And say that 15 times fast. So the reason that it's called CRISPR is because people actually discovered these things way before they had any idea what they were doing. And I kind of love this example because it's really kind of genomics-enabled science. It was literally genome sequencing, how people found CRISPR in the first place, and then figured out how it worked later. For most of the things we've talked about in this class, you have something that is happening, a function, and then you figure out how that function was derived genetically. So this is really flipping that on its head. The original use of CRISPR, and when I say original, a historical, evolutionary-wise, CRISPR is really a defense mechanism against virus infection in bacteria and archaea. So how does it work? You have an infection now by a DNA virus. We talk about RNAi as protection against RNA viruses. DNA viruses, the vast majority of viruses that infect bacteria are DNA viruses. These DNA viruses will infect, they'll put their genome inside the cell. This is exactly how DNA as being the genetic material. Chase and Avery, way, way back. So we have this DNA that gets chopped into smaller pieces, and these smaller pieces get put into the bacterial genome. and they actually provide a memory in this bacterial genome of previous infections that have taken place. Then this memory bank, basically, is transcribed, chopped into smaller pieces, and each of these smaller pieces now remembers that, hey, there was this nasty DNA virus that infected us a while ago. We remember that it's here. We'll get rid of it. And this is very similar to our own adaptive immune system, except we're using antibodies for this process. But here it's a DNA-based immunity process. So I'll talk a little bit about the history, how it works, and then hopefully at the end we'll have a chance to talk about some of the amazing functions that people are now using this system for. So as I mentioned, this has been discovered through genomic sequencing. It's actually discovered through genomic sequencing by this guy down here, Yoshi Ishino, in 1987, who was sequencing a gene in E. coli. And he sequenced the gene in E. coli, which in 87 was actually not easy to do. And then he had this really strange sequence right next to it. And he mentioned it in his paper. And what that strange sequence was it had these repeated sequences spread out about every 30 nucleotides with sequences that didn't really match anything else. And these were called the spacer sequences. So there were these repeat units and then these spacer sequences which were found. Now these repeat units were really interesting because they were these palindromic repeat sequences. And palindrome, reads the same way on the opposite strands. What that means is that these can actually form base pair structures with each other. And so this is, in fact, the repeat structure from E. coli. You have 5' to 3' continues, 3' to 5', and so you can base pair here. Turns out that almost all bacteria and archaea have these present in their genomes. But nobody really had an idea what they were doing. So a couple of years later, actually about 13 years later, these other genes were discovered. And these are called the CRISPR-associated genes. So the CRISPR, again, cluster of regularly interspersed palindromic repeats. So people were really interested in these repeat sequences, right? You've got a repeat, 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 repeat. And so I actually found one of these in one of the plasmid genomes that I was sequencing, this funky repeat sequence. And actually we called it an SRSR for sequence repeat. So repeats, we thought it was something important about these repeat sequences. And we thought it had something to do with DNA repair or combination, or maybe in the case of the plasmids, making sure each of the plasmids ended up in the right cell. But about then, and actually in the mid-2000s, people started looking at these genes that were next to all of these CRISPR arrays. They were pretty well conserved. And I was at a meeting in 2006 where Eugene Koonin, he calls himself actually a computational biologist at the NCBI, National Center for Bioinformatic Information, at the National Library of Medicine at the NIH. This guy has like 1,000 papers. It's absolutely insane. Ridiculously smart guy. Also a great marathon runner. But at this presentation, I was talking about our virus sequences, and he right before me said, actually, I see these genes right next to these CRISPR loci, and I think it might actually be an RNA interference-like kind of process, because he looked at these genes and he said, looks like they might have something similar to some RNA nucleases. Maybe there's something like some of these Argonaute proteins that are associated there. I think there's something to do with that. And at about the same time, Francis Mojica, who is a Spanish microbiologist, noticed that, these repeat sequences are really interesting, but way more interesting are these spacer sequences, which seem to match either virus sequences, plasmid sequences, etc. So it was like, okay, we're going to ignore these repeats for the time being. Let's get interested in what's going on with these spacer sequences. And so it was really the combination of Eugene Koonin and Francis Mojica who sort of said, okay, I think it's kind of an RNA interference mechanism, and we've got all of these sequences that are matching these extra chromosomal elements. Probably what's going on here is some kind of antivirus defense mechanism. And sort of the Everything kind of blew up in 2007. There was a paper in Science by Rudolf Barangou and some collaborators. They were literally looking at virus infections in Streptococcus thermophilus. Where do you know Streptococcus thermophilus from? So no, not actually from strep throat. A little yogurt pot, active cultures. So Strep thermophilus is one of the major bacteria that's important in yogurt and cheese production. What happens if you have viruses that infect your yogurt and cheese production bacteria? Big problems, because they're all dead and you don't make your yogurt or cheese. So Rudolf Barangou was actually working for Danisco, which also makes Dannon yogurt, at the time, trying to figure out what the problem was with virus infections and resistance against virus infections in these Strep thermophilus cultures. And what they showed really nicely was that it was actually a match that was present in one of these spacer sequences. If you had a match to a virus, that strain was resistant to that virus. If you got rid of the match, then it was susceptible to that virus, and you could mix and match these things, which is a really amazing breakthrough. A few other things that they found, and I just wanted to point this out right now, is that in the virus genome, you had something that matched your CRISPR RNA, but in the virus genome, you also have this extra sequence right here, what's called the PAM protein, or the protospacer, which is just this piece of DNA in the bacterial genome, next to a protospacer adjacent motif. And it turns out that having this sequence right here, which is different than the spacer you have in your own genome, is absolutely critical to make sure that you don't chew up your own genome. Because if you have a mechanism where you're chopping up any DNA that comes inside the cell, and it's based on a sequence that you have in your own genome, you better be really careful you don't chop up your own genome, and you just chop up the viral genomes, or the external chromosomal elements. So that's what this PAM is really all about here. So now I'd like to step through how this process actually works in a bacterial system. Basically 3 steps: adaptation, expression, and interference. Just what we had before. Again, the figure from your textbook, this is a slightly different one here. So the adaptation process is taking a piece of DNA from this incoming virus, again, right next to one of these protospacer adjacent motifs, and incorporating it into a CRISPR locus. This CRISPR locus, again, has repeat sequences. It's got all these spacers in it, and you're bringing in a new spacer. One other thing that I hadn't talked about so far, and that is all of these CRISPR loci also have a leader sequence that's associated with them. This leader sequence basically plays 2 roles. One is it's important for adding more of these spacers, but just as important, it also has a very strong promoter. And that very strong promoter is going to lead to transcription of this whole locus. When you make an RNA from this locus, you end up with this pre-CRISPR RNA, and that then gets broken down, and you end up with effectors. But let's first look at adaptation. How does adaptation work? Adaptation works. There's actually a whole bunch of different kinds of CRISPRs. We're not going to get into them because we don't have time to. But the one that most people care about is this Cas9 CRISPR, because that's what's used for human genome editing. The Cas9 protein, together with its own RNA, and this is an RNA, a separate RNA. It's called the tracer RNA, also present in this leader sequence. That then will bind to PAM sequences and some other external double-stranded DNA that's coming inside the cell. Once that happens, it activates these 2 other proteins, Cas1 and Cas2, that basically serve as an integrase. So they will bind to the protospacer, bring it to a CRISPR locus, and with the leader and the next spacer, duplicate this repeat sequence. And actually functions a lot like integration tyrosine recombinase that we've talked about before in terms of site-specific recombination, and puts this spacer in between 2 repeat structures. And so first binding, and again, really important here, that PAM sequence. If you didn't have a PAM sequence, you're not going to be cut. You have to have this PAM sequence in order to have a protospacer that's going to get integrated. Once we have that, then we have expression, again, from this leader promoter sequence, giving you your pre-CRISPR RNA. That is then chopped up into smaller pieces. So each of these palindromic repeats, not surprisingly, folds up into that secondary structure. And then your CRISPR RNA then gets chopped into a piece that has a spacer, but also has this sequence and now structure, which is going to interact with the effector proteins, particularly the Cas9 protein, which will lead to your final interference step. Interference step, again, that's chopping up the double-stranded RNA once it gets inside the cell to protect the cell from a secondary infection. This works in lots of different ways in lots of different CRISPR systems, but the one, again, that is mostly being used now is Cas9. Cas9 has this protein sequence, which has a couple of nuclease domains that are associated with it, which are going to cut on both strands of the DNA. Also interacts with this PAM sequence through a separate RNA. This is that tracer RNA. The tracer RNA, again, is something that helps bind to PAM sequences and gets your spacer RNA associated with this process. One of the things that people have done now more recently is they've turned it into what's called a single guide RNA. You see the 5' end and the 3' end here are really close to each other. So just through fun molecular biology tools, you can connect the two of them to each other and just make this as one RNA. So it's an RNA that associates with Cas9 and an RNA which is going to give you specificity for breaking down the double-stranded DNA once it's bound. So that's our process: adaptation, Cas9, Cas1, and Cas2, biogenesis, which is transcription and nuclease activity. Then it gets assembled into an effector complex, which will chop both double strands of the DNA. This was all done before two very, very intelligent women came up with the idea that, oh, well, if this is happening in bacteria, maybe we can get it to work in eukaryotic cells. So this is Jennifer Doudna and Emmanuel Charpentier, who got together and basically said, well, maybe we can take these guide RNAs, which have sequences in them, that we can design to be anything, and it could be complementary to eukaryotic sequences. Now maybe we can start to use this to modify eukaryotic RNA. Now, eukaryotes don't have any of these things. They don't have Cas9. They have none of these other CRISPR systems. So it's all stuff which can be brought in from external to the cell and then brought into the cell and then have the activity. And so this is a now very short list, it may not look like it, of some of the applications which CRISPR-Cas9 is being used for. Originally, it was used for deletions. And why it was used for deletions? Basically, what happens, you have a double-stranded break in your DNA. How did double-stranded breaks get repaired? Way back when. Two basic mechanisms: non-homologous end joining and homologous recombination. So, non-homologous end joining, what does that do? Chews in the ends and puts them back together. What happens when you chew in ends, particularly if you have a coding region in your DNA? It's going to disrupt that gene. And this is actually the original gene disruption processes with CRISPR-Cas9 was literally chopping the DNA and letting the non-homologous end joining machinery chew in at the ends and put it back together. This is nice and well and good, but as we talked about when we talked about DNA repair, it's kind of the quick and dirty approach, because you chew in at the ends and then try and put those ends back together. Who knows what you're going to get? Homologous recombination is a way better way to go, because then you end up repairing that double-stranded break with a homologous chromosome, or for that matter, an extra piece of homologous DNA, which you happen to put into the cell at the same time you put in your CRISPR-Cas9. And so that's how people are actually doing gene replacement, is putting in an extra piece of DNA together with the CRISPR-Cas9, and you can get gene replacement. And this is really exciting in terms of thinking about gene therapy and CAR T cells, all kinds of genetic modifications. You can make a genetic modification in a really specific place in a genome that could turn out to be really, really useful. So these are just using the regular CRISPR-Cas9. Lots of people, I think there's a spoof journal that's called the CRISPR journal, the latest journal to come out of the Nature process, and all of these things, they're all CRISPR all the time. But one of the more important things that I got out of that is you can actually now take the Cas9 protein and get rid of the 2 nuclease activities on both strands. And then this is basically a way to get any protein you want anywhere in the genome. And it turns out to work out really well even in the presence of chromatin, which is potentially why a lot of the other mechanisms haven't worked very well. So if you put in CRISPR-Cas9 with the guide RNA with an inactive form of the actual Cas9, you can fuse it to all kinds of other things. So activators, so if you want to activate a particular gene, you put in an activator fused to your CRISPR-Cas9, get it to bind to wherever you want it to, it'll activate the gene right next to it. Do the same thing with repression. And some of the more exciting things that have just been coming up really recently is you can literally put an editor protein. And it turns out that there are DNA editors as well as RNA editors. You can attach those to CRISPR-Cas9, and it will bring that gene editor to exactly the right place in the genome. And so you could maybe actually reverse some genetic errors that have taken place, just literally change the individual nucleotide at some place in the genome and get that to work. So, et cetera, et cetera, et cetera. We could go through many, many of these, but I just wanted to talk about 2 final aspects about this. These are 2 papers which I just pulled up, recent ones, one from last month, one from last year. Not surprisingly, if you have this wonderful defense mechanism that your cell has for protecting against viruses, they've been evolving as well as the cells. So the viruses have actually come up with mechanisms to block how CRISPR works. So anti-CRISPR proteins. And so this is just an example of some of the anti-CRISPR proteins that were found in some viruses. They block a slightly different mechanism of how CRISPRs work, but basically in the same way. Here's another system where now they've taken these defective Cas9, so these little yellow dots here represent mutations that mean it doesn't have nuclease activity anymore, and then they've hooked it up to a transcriptional activation domain. This is the VPR transcriptional activation domain. So just by having this guide RNA together with your inactive Cas9 connected to an activator, you can activate whatever gene wherever you want it to be. You can put a repressing domain, same kind of thing, specific place, and then it's the presence and absence of this CRISPR, anti-CRISPR protein. You can change all of these things. So then this is great, right? So we can modify genomes, et cetera, et cetera. One big problem. CRISPR-Cas9, particularly Cas9, comes from bacteria. Some of these bacteria, like Streptococcus thermophilus, in our yogurt. So turns out that most people have antibodies to Cas9 proteins, and most of the Cas9 proteins that are generally being used. So actually putting this into someone may actually be a real problem, because it may not actually work at all, maybe not too different from these siRNAs as well. Mostly what people are looking at right now is to try and do all of these gene modifications by taking cells out of a patient, modifying them, and then putting them back into cells. So again, we could spend a whole term, be nice if we had those courses, talking about CRISPR and all the various different modifications. I do know that Dr. Bartlett will be talking about this quite a bit in his recombinant DNA techniques class, the lecture part of that next term. Friday is review. Everybody gets 2 clicker questions because I forgot to add them here. 2 clicker points, I should say. I'll post those questions so you know what they are. And review on Friday, exam on Tuesday.